Oh, thank you very much for that introduction, Rebecca, and thanks for the invitation. I'm really happy to be here. Um, so I'm gonna just start by talking about some of our adventures in computational biophysics in my lab. We work in these 4 different areas. So as Rebecca mentioned, we focus a lot on simulations of key biological processes, including protein binding. We also rationally design protein conformational switches, and a natural switch includes the spike on the surface of the coronavirus. And then we develop ways of extending the timescales of simulations, including the weighted ensemble strategy. And then finally, we develop more accurate force fields simulation models for simulations, including the AMBER FF15 IPQ and IPQM force fields that are used for protein mimetics. So today I'm going to talk about just these first 3 adventures here. And our main instrument in the lab is a computational microscope involving molecular dynamics simulations. So this, as some of you are familiar with, are simulations of how atoms move according to physics and chemistry. Shown here is the binding process of Barnase and bar star, 2 proteins diffusing towards each other in a box of water molecules. So these flickering molecules are the water molecules. So when I was a graduate student with Peter Coleman, I was really excited about this Gabdeling and Wade paper that came out of Rebecca Wade's group involving the binding of the Brownian dynamics simulations of these this binding process, and wanted to figure out how to capture the conformational changes of these proteins when they bind one another. And wanted to do this without biasing the dynamics so that we could go after the kinetics. However, back then when I was a grad student, we didn't figure out an appropriate method for doing this, and binding processes are slower than your typical molecular dynamics simulations. And these are the sorts of timescales that we're dealing with for these simulations. So starting from femtosecond time steps to capture the fastest motions, which are the bond vibrations. On a typical cluster, we can capture up to a microsecond, which can capture some motions on proteins such as loop motions and hinge bending, but we're missing all these interesting functional motions of proteins such as folding and large-scale conformational transitions. With the Anton special supercomputer, special purpose supercomputer, as well as GPUs, we might be able to reach milliseconds for certain small systems. But we still want to be able to access these motions beyond the millisecond timescale. So how do we do that? So this is the overview of my talk today. Is I'm going to start by just talking about my journey in terms of extending the timescales of MD, starting with these early simulations of protein binding when I first started up my lab, going into the weighted ensemble strategy, and then presenting 2 applications recently of weighted ensemble, including chemical reactions and the opening of the spike protein. And then finally, I'm going to wrap up with some recent advances that we've made to the weighted ensemble strategy. So I'm going to start with these early efforts of ours in protein binding. And to start, let's just give a simple illustration of a protein binding process, which often involves 2 steps. So this is just a schematic diagram of 2 proteins in which these binding partners are initially dissociated, and the partners associate to form an encounter complex, which is close to the right orientation, but not quite. And then this encounter complex would rearrange to the final state. And it's not usually this smooth. It's a more stochastic process, just like the initial collisions of those proteins. So we initially focused on this second step, focusing on the collision of a peptide fragment with a protein to form a beta sheet. So this model peptide protein system in which a peptide fragment of the signaling protein HNFK in yellow is forming an intermolecular beta sheet with this Ki67 FHA cancer marker. This was a structure solved by Angela Gronenborn's lab at University of Pittsburgh. And we wanted to look at how if the peptide fragment collides such that the beta sheet is misaligned, how does the beta sheet rearrange to the correct alignment? So how does nature correct for that mistake in collision? And this was a paper that came out in 2011, our early efforts involving two co-first authors that eventually married each other years later after being in my lab. And so a closer look at this beta sheet here. So this is the peptide beta strand with the various amino acids, and this is the protein beta strand. And the dotted lines indicate the hydrogen bonds between the strands to form the beta sheet. And then I've also colored these hydrophobic residues in green. And so something interesting about these beta sheets is that these hydrophobic residues line up with each other across that between the strands. And so that really limits how the beta sheets can be misaligned. And so the two strands could be misaligned by only shifting up or down by two residues as dictated by the alignment of these green residues. So this is where it would be shifted two residues down, and then this misalignment would be where they are shifted upwards by two residues. And so you see that there's still this alignment of at least one pair of green residues here. And we wanted to be able to simulate from these two possible misaligned states, the rearrangement to the correctly aligned state. So how might we do this? It's challenging to simulate because they take a long time. So why is that? Well, fortunately, these processes are rare but relatively fast. So meaning that if you started a number of simulations, which are indicated by these spheres from the initial misaligned state, and every so often you'll get a lucky transition over the free energy barrier to the target aligned state. And so something that's been done in the early days of distributed computing with the Folding at Home project, for instance, is to run a large number of these simulations in the initial state with the hope of increasing the likelihood of capturing these lucky transitions. So in fact, this was the strategy that I used when I first started up my lab at the University of Pittsburgh. To revisit these protein binding processes. So how did we simulate the rearrangement? We ran hundreds of short simulations, so 20 to 40 nanoseconds, starting from each misaligned state. And we extended 20 of these simulations to 300 nanoseconds. So at the time, this would take about a month. And so it was already really involved. And then out of these 10, there were 3 successful rearrangements. And so this is a movie of one of these rearrangements. And it was a common mechanism that involved the crawling of these aromatic residues. So in particular, this phenylalanine colored in green on the receptor, and then this phenylalanine on the yellow peptide. Those are the two to look out for. So the green phenylalanine essentially acts as a gate that's going to swing out to allow this yellow phenylalanine to anchor into a transient pocket. And this is the way this yellow peptide moves from this position where it's shifted upwards by 2 residues down to the correct alignment. So I'm just going to show this movie here. So in gray is this receptor, and you're looking out for this green phenylalanine to swinging out, and this swinging out motion is actually intrinsic to the receptor dynamics. Okay, so it's just waiting for it to swing out. Yeah, so it's only when it swings out where this pocket gets revealed, and then this phenylalanine on the yellow peptide anchors, and that anchoring allows everything else to fall into place to the correct alignment. And one interesting that we found is that when we tried targeted MD, in which we're artificially accelerating the dynamics towards the target state, we didn't see this crawling mechanism. And so this is just an early indication to us that it can be worth it to generate these pathways in which the dynamics are not biased to get these rich mechanism details. So then we went further and examined, well, why did we just get these three successful rearrangements? And so the problem with these standard simulations, even when we run a large number of them, is they're still spending too much time in the initial state and not enough time on these transitions between the stable states, which is what we're actually interested in. So those are the functionally relevant motions. And so how can we be even smarter without using any external forces? So how can we focus the computing time on just these transitions? And so the weighted ensemble strategy is one way of doing this. There are many other ways of doing this. So this was a strategy, as Rebecca said, that was developed in 1996. And it was by Gary Huber and Sante Kim, and then rediscovered about a decade later by Daniel Zuckerman. And Dan and I have collaborated for more than a decade on making this strategy even more efficient and accessible. So in this strategy, we typically divide a progress coordinate toward the target state into bins. We then run many short trajectories in parallel, and then at fixed regular time intervals, we enrich for success by replicating trajectories or splitting trajectories that have made transitions to empty bins. And then every so often we save computing time by terminating trajectories that have not made transitions. So there's other splitting strategies like this, including adaptive sampling, adaptive multi-level splitting, forward flex sampling, etc. And so the ideal outcome from this weighted ensemble strategy is that it generates continuous pathways that are unbiased in dynamics with orders of magnitude less computing time than standard MD. And this efficiency scales exponentially with the free energy barrier, and also the target states do not need to be strictly defined in advance as we're running the simulation. And finally, pathways are the mechanism. So generating these pathways from the initial state to the target state, that's the most direct way of analyzing mechanism. And that includes all the states, including transient states that are too fleeting for the experiments to capture. Okay, but there's no free lunch with any of these enhanced sampling approaches. So with weighted ensemble, the main caveat is that you might always miss these slow coordinates that are orthogonal to the progress coordinate that we're using for the weighted ensemble simulation. However, there is this flexibility that progress coordinates can be switched on the fly as the weights of the trajectories that we keep track of rigorously are not linked to the progress coordinates. They're completely independent of them. So the study that convinced my lab to proceed with weighted ensemble MD was a number of benchmark systems that we used for molecular association. So starting with association of methane-methane in explicit solvent, and then increasing in complexity. And all of these systems are, all of these processes are processes that can be run down to the ground with brute force standard simulation. So the next one was association of NaCl, and then the association methane benzene, and then finally the most complicated was the association of a potassium ion with crown ether, flexible crown ether in explicit solvent. So in all cases, weighted ensemble is much faster than standard MD in estimating the rate constant for association. We also looked at the distributions of times for transitions from the initial to target state. So that's what these transition event durations are. So the red distributions here are for weighted ensemble. Blue distributions are from standard brute force MD. So You can see that the red distributions are essentially the same as the blue distributions, except for this last system where for the crown ether, actually brute force didn't entirely converge. Weighted ensemble was better resolved. And then these numbers in these upper right-hand corners are the speed up. So 7.5 times means that we can use 7.5 times less computing time. And this efficiency increases as the free energy barrier to association increases. So this last number here, orders of magnitude greater efficiency, which is what excited me about this strategy. So my lab scaled up this strategy to tackle complex systems and processes developing this WESPA software package. So it is scalable out to hundreds of GPUs or thousands of CPU cores. And the weighted ensemble strategy is general, meaning that it can be used with any kind of stochastic dynamics, including Brownian dynamics, Monte Carlo, even dynamics and systems biology packages with non-spatial systems. And it has a modular design. And this is where you can find the package. And we've applied this strategy at multiple scales. So working up to simulating protein binding at the atomistic level with explicit solvent, we started out with very coarse grain models of Barney's bar star, and then gradually worked up to protein peptide binding in implicit solvent. And finally, this is the system that I showed early on in the talk with Barney's and bar star, a classic system for studying protein-protein binding. And so this is when we were finally able to generate hundreds of pathways for protein binding with fully flexible proteins. And this was done in about 10 days. And these days you could do this with 10 days using 16 GPUs. Okay, so the movie that I showed you earlier was from this work. And then finally, the weighted ensemble community is continued to expand. So these are just some examples among dozens of groups, including early efforts by Dan Zuckerman's lab at the systems biology level. And this paper here in 2018 that involves Ariana Nunez Alves in the Aranti lab at the time at the atomistic level involving protein ligand and binding. And most recently, there have been efforts from the Fujisaki lab and D.E. Shaw research. Okay, so now I'm going to go into some recent weighted ensemble applications, starting with simulations of chemical reactions. So this is a point where I've been able to connect some of what I've been teaching in my undergraduate quantum mechanics course. So this was an effort revisiting a textbook reaction involving the azide clock. So this presumably a diffusion controlled reaction of an azide ion adding to this carbocation, which is a rather stable carbocation due to all these three phenyl rings. So this is a methyl phenyl based carbocation. When you have a substituent here, this R group, depending on the R group substituent, the reaction can be anywhere from hundreds of nanoseconds to microseconds. And this was a project that involved Anthony Boghetti, graduate student in my lab, and a former student, Matthew Zwier, who's currently a professor at Drake University. So we wanted to go after generating complete pathways of these chemical reactions in explicit solvent. And so not just locating the transition states, but including all of the transient states as well as the stable states. We focused on 3 cations with different substituents. So the slowest reaction involved a methoxy group at the R position. So that's an electron donating group. And then the fastest reaction involved the CF3 substituent, so an electron withdrawing substituent. So these reactions you'll find in your favorite physical organic chemistry textbook as an example of how electron donating and electron withdrawing substituents might impact the reaction. And so we're showing here the Mulliken atomic charge at this PM60 semi-empirical level of theory. You can see differences in the extent of positive charge in this central carbon here. And also back in college organic chemistry, we learned that a charge could be delocalized as illustrated by resonance structures. So these are resonance structures for that unsubstituted carbocation. You have the positive charge at the central carbon here, which is what we think of as the target product when the azide anion adds to this central carbon. And then these are some alternate positions where alternate products might form, shown in blue here. How did we do this? The simulation model involved a hybrid quantum mechanical molecular mechanics model. So as a first step in this direction, we used a semi-empirical level of theory, PM6D, for just the reactants, and then treated all of the solvent, which is a mixture of water molecules and acetonitrile, classically with your usual force field parameters. And then we ran the simulations in 2 stages. So first we ran a standard simulation to generate 50 orientations of the unassociated reactants. And then we ran a weighted ensemble simulation, actually multiple weighted ensemble simulations from randomly selected orientations. And what you're seeing on the left here is the location of all these different azide anions relative to the carbocation. So we were able to generate hundreds of complete reaction pathways. This is a probability distribution as a function of the progress coordinate that we used for the weighted ensemble simulations. So this was a 2-dimensional coordinate tracking the distance between the nitrogen on the anion to the to the central carbon distance, and then the y-axis was tracking the distance to the carbons on the phenyl rings. Okay, and so this would be our products, and then our reactants would start over here, and we're coloring based on a free energy scale in terms of KT. So yellow here is our target product where the azide has added to the central carbon. And in blue here, the blue star, that indicates an alternate product where the azide has added to one of the phenyl carbons. And what I'm showing along these lines here, so the yellow is a representative continuous pathway that was generated to the target product, and in blue is a representative pathway to the alternate product. So let's look at a representative pathway that we capture. So again, these were simulations in explicit solvent, and here's the azide anion, and we're going to see it adding to the central carbon on this carbocation, and in the lower left-hand corner is the probability of each conformation. So they're very rare probabilities. And what about the reaction kinetics? So we were able to calculate rate constants that were reasonably converged. So in blue are our estimates from weighted ensemble. In gray are the experimentally measured values, which is back from 1991. And we can see that they're in reasonable agreement with experiment, especially since the semi-empirical level of theory was used for the quantum mechanics. So these 2 cations, so the unsubstituted and the electron withdrawing substituent cation, are close to the diffusion controlled limit, while this methoxy substituted cation is a bit slower. And we also had enough sampling to estimate the % collisions that are productive for forming the target state. So this % of productive collisions to the target state gradually increases, going from the electron donating to the electron withdrawing substituent. And we were able to detect alternate products that were previously not observed in experiments by looking at the % azide additions to various sites. And so the percentage is indicated by the sizes of these pink spheres. So the larger the sphere, the greater greater the percentage, and we've overlaid these spheres on top of the Mulliken atomic charge coloring of these cations. So for the unsubstituted cation, most of the additions are happening at the central carbon. When we move to the electron withdrawing substituent cation, alternate positions are at these unsubstituted rings rather than the substituted rings. So the electron withdrawing substituent here prevents additions to sites on the substituted ring. When we move to the methoxy substituted cation, it's mostly adding to this carbon that's attached to the methoxy substituent rather than the central carbon. And that's also due to resonance stabilization. And there's no additions to these unsubstituted rings. To summarize this part of the talk, the weighted ensemble strategy generated pathways, complete pathways for these azide clock reaction. And this was done in explicit solvent. And we got some reasonable rate constants as validation of these simulations. Our simulations revealed some insights about mechanism that weren't apparent from the experiments, such as certain alternate products, and we're still in the process of determining some important dynamical features for the mechanism. And this is kind of a proof of principle that demonstrates that weighted ensemble can be used to enable us to simulate reactions on the microsecond timescale, up to the microsecond timescale with a similarly sized system, so less than 50 atoms in the quantum mechanical region. All right, so now I'm going to move on to the other extreme where we've tried to simulate, we've simulated the opening of the spike protein on the surface of the SARS-CoV-2 virus. So this is the largest system that we've yet simulated. And as Rebecca mentioned earlier, this was part of a massive collaboration involving 28 members, 10 institutions, 4 supercomputing centers that got the Gordon Bell Special Prize. And this was, our prize was dedicated to the memory of Klaus Schulten. So we got involved in this collaboration. So Anthony Boghetti and I, my grad student and I, with the team at UCSD that was led by Romy Amaro. And so it was specifically this team here, so the WE team. The team that worked on the simulation involved both members from my lab, as well as Romy Amaro and Andy McCammon's lab. And this was in collaboration with experimental work that was led by Jason McClellan. The person that got us into this collaboration was a graduate student, very motivated graduate student, Tara Stein, who is joint between the Burkert and McCammon labs. And so she approached my lab with an interest in simulating the opening of the spike proteins that are on the surface of the coronavirus. And these spike proteins must open in order to latch onto human cells in order to fuse with them and then ultimately infect them. So we decided to focus on just this head region of the spike protein. So focusing on the minimal part that opens. And this is a process that could take years to simulate with standard MD simulations because it is a second timescale process. So this is where an enhanced sampling approach is sorely needed. Our simulation model involved the Charm 36 force field, tip 3P explicit water, and we simulated using this model of the closed spike, fully glycosylated. The glycans, the sugars are indicated by these dark blue attachments. And the opening process involves this receptor binding domain colored in cyan, which is going to move from down to an up position. So we tried to captured the spike opening process in 2 stages using a 2-stage simulation procedure. So first, we ran a weighted ensemble simulation to extensively sample the conformations of the closed state. And this is an important step to ensure that we have a well-equilibrated initial state. And then we ran 2 weighted ensemble simulations of spike opening, starting from the equilibrated closed conformations. So our simulations did capture spike opening. And what was interesting is that we also captured opening beyond what was observed experimentally. So this is a distribution of the conformations that we captured as a function of the progress coordinate that was used for the weighted ensemble simulation. So our progress coordinate is intended to capture the slowest relevant motion. Along the y-axis is the root mean square deviation of that receptor binding domain up here relative to its up position, which is our target state. And then the x-axis is the distance between the center of mass points of the receptor binding domain and the core domain. And we're coloring all these dots based on the percent solvent accessibility of that RBD, the receptor binding domain. So we're going from this down position, which is that closed state of the spike protein, down to the up position, which is when it is open, but we also sampled a beyond open state, so a further open state that wasn't observed experimentally. And this further open state actually corresponds to the spike conformation that is bound to the human receptor ACE2, so the cryo-EM structure. Okay, so I can show you a movie of a representative pathway that we captured, and this pathway revealed a glycan gate at the position of residue N343 that actually controls the opening of the spike. So this is a top view of the spike, where again, the dark blue is the glycosylation or sugar coating of the spike protein. And at the atomic level, we can see that one of these sugars or glycans that is highlighted in magenta pushes up this receptor binding domain in cyan from the down to the up position. Okay, so I'm going to start this simulation. Hope it doesn't choke. See if I could get this to load. Sorry about this. Okay, so you're going to see this magenta residue pushing up this RBD domain. And it's sort of a hand jive motion that it pushes it up, and eventually this receptor binding domain ends up in not just this up state, but that further open state that we were able to observe in the simulations. So in other words, this glycan in magenta is acting as a gate that controls the opening of the spike protein, which was previously unidentified. And experimentally, the importance of the glycan in the opening process was validated by mutating the residue N43 to an alanine, and that reduces binding to the human receptor ACE2 by 56%. And here's the preprint for more details about the mechanism. So we were able to generate 310 pathways for the spike opening process, and this was using 100 NVIDIA V100 GPUs on a supercomputer. So really an unusual amount of computing time that was given to us for this coronavirus research. And this was for a massive system of around 500,000 atoms using WESPA, coordinating the trajectories that, that were run by Amber so that we could take advantage of the GPU acceleration. So generating the 310 pathways, that took us 40 days using 100 GPUs at a time. If we were to generate just one pathway, that took about 15 days. And since this is a seconds time scale process, standard MD would have required hundreds of years to generate just one event. And so this is indicating that weighted ensemble MD is at least 27,000-fold more efficient than standard MD. And I just want to also mention that in another study by Unfolding at Home by Greg Bowman's team, they were also able to generate opening events of the spike protein. And this involved adaptive sampling, which is another splitting strategy that replicates promising trajectories. And that demonstrates the power of these splitting strategies in efficiently capturing these slow processes. All right, so and weighted ensemble has not reached its full potential. So I'm going to talk about some advances that have been made to it to make it even more efficient in a minute. But first, let me summarize this part of the talk. So we were able to generate pathways for a seconds timescale process for this massive protein. So this is quite a high watermark for these ensemble MD simulations. And our simulations revealed spike opening beyond what was observed experimentally, as well as this glycan gate that controls the opening process. So that gates the switching process of this natural protein. And then finally, our pathways include transient states that can be used to aid drug discovery efforts. And this includes those further open states. All right, so I wanted to just wrap up here now with recent advances in weighted ensemble MD. So one scheme that my lab has recently developed is for adaptively placing the bins along a progress coordinate instead of just manually using fixed spinning schemes that we adjust on the fly. So this is a minimal scheme that works by tracking the positions of the trailing and leading walkers along, so the trailing and leading trajectories along the progress coordinate, as well as identifying bottleneck trajectories in regions where the probability drops off very quickly. And then we adapt the bin positions based on the positions of the trailing, leading walkers, and bottleneck trajectories to get more efficiently over a barrier. And we showed that this scheme can surmount a large barrier for a toy model system, so this 35 kT barrier, if we use a manual fixed spinning scheme and track the probability distribution as a function of weighted ensemble iterations and the position along this coordinate, so this one-dimensional coordinate x. All of the probability is in the initial state. If we use the adaptive binning scheme, we get more even coverage all throughout this x coordinate. And this is because of tracking early identification of these bottleneck trajectories. And another advance that we've made is a scheme for estimating rate constants from shorter trajectories. And this made the cover of the Journal of Chemical Physics recently. And so normally when we estimate a rate constant, there's this transient region for the rate constant as a function of time for the simulation. In this transient region, we usually leave out in our calculation and then only focus on the part that has leveled off, that has reached a steady state. So we could actually utilize the full simulation, including that transient region, if we incorporate the distribution of transition times that make it so the times for making it from the initial to the target states. So using this equation here, where we're normalizing essentially by this distribution of transition times. And this is akin to making use of the times for the initial runners that finish a race in a cross-country race. So using this scheme, which we called the rate event duration scheme, the red scheme. We applied this to the Barney's bar star simulation, so our protein-protein binding simulation using weighted ensemble. So in blue is the rate constant estimate from the standard weighted ensemble scheme. And then in red is from our red scheme. So for reference, the gray line shows the experimentally measured value for the rate constant. And we show that with the red, we do get to this leveled off region earlier. So it's about 1.3 fold more efficient in estimating the rate constant for protein-protein binding. And finally, I just want to close by talking about these timescales again. So weighted ensemble MD greatly extends the timescale that's accessible to molecular dynamics simulations. So as I mentioned earlier in my talk, standard MD can allow us to access maybe up to a millisecond timescale, especially using GPUs for certain small systems. Weighted ensemble has enabled up to the seconds timescale and for a massive protein. Right, so I want to close by showing you a picture of my lab, and want to thank also the WESPA software developers, which is maintained by more than just my lab. It's a community of developers, so including Matt Zwier, who's a former student in my lab, the Zuckerman lab, as well as OpenEye Scientific and others. And then I just want to mention that our next version of WESPA is coming this summer. So inspired by our tackling of the massive spike protein, we've developed a number of features that will facilitate handling of larger data sets, automated binning, there's new analysis tools, ways of calculating rate constants from pre-steady state simulations and more. And thank you for tuning in today. And I'm happy to take any questions that you have.